Hello everyone, welcome back to Microbiome Solutions. Today we're going to discuss about rhabdovirus or virus causing rabies infection. So they belong to the family Rhabdoviridae, and under Rhabdoviridae there are two genus. First one is vesiculovirus, which contains vesicular stomatitis virus, and Lyssa virus, where rabies virus is there. So Rhabdoviridae under genus Lysovirus, there is rabies virus. So rabies virus is a bullet-shaped, enveloped, single-stranded RNA virus which contains a lipoprotein envelope which has glycoprotein spikes. And nucleocapsid contains RNA-dependent RNA polymerase, and this virus is sensitive to alcohol, soap, detergents, and ether. Also, they get inactivated by phenol, formalin, BPL, UV, and sunlight. These are the common features of rabies viruses. So you can see the picture of rabies virus. As I have mentioned, it is bullet shaped and it has got an RNA genome. And inside the RNA genome, there is RNA dependent RNA polymerase, and covering the RNA dependent RNA polymerase and the RNA genome, there is a nucleocapsid. So the nucleocapsid is helical in nature. The shape of nucleocapsid is helical in nature, and covering the helico nucleocapsid, there is an envelope. An envelope has got a G glycoprotein spike. This is the structure of rhabdovirus. Moving on to the pathogenesis of Rhabdoviridae, as we all know, the causative agent is rabid dog, and mode of infection is bite of a rabid dog. Rarely, it can transfer through licks and aerosols. Infection in human manifests as hydrophobia, phobia to water. So the pathogenesis starts with the bite of a rabid dog, and virus present in the saliva of the rabid dog will be deposited in the wound which is generated due to the bite. Multiply locally in the connective tissues and muscles and nerves. So the virus is entering from the saliva of the animal through the wound, and it multiplies in the local connective tissues, muscles and nerves. So after the multiplication, it will penetrate the local nerve endings and travels in axoplasm towards the spinal cord and then to brain. So later it spreads from axon to neuronal bodies and progressively up to spinal cord. And from the spinal cord, it reaches brain and multiply. So first multiplication will happen locally and second multiplication will happen once it reaches the brain. And from the brain, it spreads centrifugally via nerve trunks which is present in brain and infects various parts of the body including salivary glands. And again it multiplies in the salivary glands. It can shed in saliva. Other than that, also it can shed in milk and urine. Can also be present in cornea and facial skin. So we say that when the bite of the rabid dog is in the face or in the neck, the patient is prone to get infected with rabies virus fast, as the multiplication, the local multiplication will be either in the neck or in the face, and it can reach brain faster. The clinical features of rabies virus: incubation period is short when the wound is on the face and hands, and it is long when it is on the leg, because the time for traveling the virus to brain is more when the bite is in leg, and it is short when the bite is in face or neck or hands. It is shorter in children than adults. Incubation period is related with the period required by the virus to reach the brain. And there are different stages in human. First stage is prodromal stage, second is acute neurological phase, then coma, and finally death. So first is the prodromal phase, where the patient will manifest with fever, headache, malaise, fatigue, and anorexia. Neuritic pain, fasciculation at the site of virus entry is also seen, and later it progressed to anxiety, depression, and insomnia. Next is the acute neurological phase where the patient will show hyperactivity with excessive saliva production, agitation, seizures, and these hyperactivities can be precipitated by external stimuli. Usually, the patient is present with hydrophobia. So, the patient will swallow dry solids, not liquid, because the patient is having phobia to water. So, water is an external stimuli which can precipitate the hyperactivities in patient, and the patient starts dreading even sight and sounds of water. Generalized convulsions may follow and can cause respiratory arrest. So once there is respiratory arrest, which can follow coma and then death. Moving on to the laboratory diagnosis of rabies viral infection. So there are two types of specimens that we can collect. One is antemortem specimen and the other is postmortem specimen. Antemortem specimens includes corneal smears, skin biopsy from the nape of neck, saliva, CSF, and postmortem impression smears of brain. Processing of the specimen can be done by antibody detection using ELISA and detection of nucleic acid by RT-PCR or dot blot assay. Virus isolation can be done using animal inoculation. Mice inoculation is preferred through intracerebral route. And brain is examined post-mortem for Nigri bodies. Nigri bodies is a cytopathic inclusion bodies produced by rabies virus. So mouse is inoculated with the patient sample intracerebrally and brain is examined post-mortem for the presence of Nigri bodies. Nigri bodies are inclusion bodies produced by rabies virus. Cell culture can be done in viro, monkey kidney cells, human diploid lung fibroblast, baby hamster kidney. Virus is identified by immunofluorescence staining with anti-sera. And postmortem diagnosis can be done to detect the presence of Nigri bodies from smears of cerebellum, brainstem, which will be stained with Steller's technique along with basic fusion and methylene blue alcohol. So these are the Nigri bodies seen in rabies viruses, especially in the cerebellum and brainstem. Postmortem sample of the rabies infected animal or human is taken to check for the presence of Nigri bodies. This is about our today's lecture. If you have any doubts, please post it in the comment section. Please do like, share and subscribe to our channel. Thank you.